Okay, for the case today, I am doing the objective on schizophrenia and also on the urinary proteins and epithelials. This isn't going to be particularly high yield for our case, so a lot of this stuff I'm just going to blaze over. I am going to use a few personal examples with the schizophrenia because my little brother has been diagnosed with it, and so I have a little bit of experience with it, and I can kind of elucidate some of the symptoms and personal examples of what that might mean. So, talking to my little brother last time I saw him, he kept insisting that he had intelligence from military satellites that dated back to 2009. And so I point that out because a lot of the symptoms of schizophrenia have to do with a disconnection from reality. Like the thoughts that you have cannot be true. And there's subcategories of these type of thoughts. For example, thoughts of grandioseness. So this may be classified as a thought of grandiose, that he has a personal ability or mission in life that is higher and above the average person. Now, the term psychosis is kind of an umbrella term, and there are a lot of causes. There is a list. I found about 10 different causes when I was looking into this kind of stuff. But in general, schizophrenia is a long-term mental disorder characterized by recurrent or persistent episodes of psychosis, and in that, it's a breakdown of relationships between the thoughts, emotions, and behaviors. And what that will lead to is your perception of reality is altered. It causes inappropriate feelings that can lead to inappropriate actions and a withdrawal from reality into delusion. Now, there's two subcategories of schizophrenia that were. One of them is not particularly recognized by the DSM-5, but there's a lot of research indicating that it is a real subcategory. In the past, there were subcategories such as paranoid schizophrenia and other subcategories that have not been supported by evidence, and so they've been removed from the DSM-5. However, some important evidence-based categories are not added yet at this time. We'll talk about that as well. So the pathophysiology, nobody really knows what's going on, and that's probably true with most neurological diseases, that the brain has so many millions of connections, it's almost impossible to know exactly how everything is firing, especially in the disease state. But the dopamine, so a lot of the neurotransmitters are implicated in the disease. For example, dopamine, glutamate, GABA, and acetylcholine. All these are neurotransmitters. And so the dopamine, all drugs with antipsychotic properties block dopamine receptors. So, for example, clozapine, it's a very weak D2 antagonist. It's the best drug for treating schizophrenia. Now, why is it important? Is because it's only a weak D2 antagonist, and it may be doing other things as well, or it may just be identifying a specific subtype of D2 receptors and blocking those. And so exactly how the drug is acting the best or better than other D2 antagonists is not exactly known. Now, the glutamate will bind to a receptor called N-methyldiaspartate, or NMDA. So these NMDA glutamate receptors, they're malfunctioning or functioning at a lower rate in general in those observed with schizophrenia, and that's diagnosed usually post-mortem. But so drugs have been developed to enhance glutamate neurotransmission, and the results have kind of been varied, and it'll depend on the person and also depend on the drug. So some glutamate receptor and agonists or glutamate agonists will work on one person and not the other, and different drugs work on different people. But there's nothing consistent. Now, GABA is one of the main inhibitors in the brain, and there's a GABA decarboxylase enzyme, and that mRNA, whenever you examine bodies post-mortem, studies have shown a decreased level of GABA decarboxylase mRNA. So the mRNA being what makes the protein, so there's presumably less protein. So if you don't have the GABA decarboxylase, you'll probably have way too much GABA activity, and so you're inhibiting a whole bunch of pathways that need to be firing. So then you have acetylcholine, which acetylcholine acts on receptors that are also sensitive to nicotine, and there's an increase in smoking activity in individuals with schizophrenia. So some people think that acetylcholine receptors may have some problems, and that's why people will smoke more often when they have schizophrenia. But the evidence behind this is that whenever you treat with nicotinic drugs, it can normalize some things, especially eye tracking. So some problems with the eyes and how they track is decreased in schizophrenia, and nicotine can actually improve those symptoms. Exactly how that works is still in investigation, however, so it may just be that having an overstimulation of those receptors is compensating for the problem that's happening somewhere else. So it may not be an acetylcholine problem at all. It's just going to require more studies to figure out. Like I was saying earlier, this deficit subtype, it's not in the DSM-5, so the manual that is the standard for how you diagnose different mental disorders and what a mental disorder is, it's not listed as a specific mental disorder in this standard book. But it's characterized, like I said, by negative symptoms, and I pointed those out. But this subtype has a stronger association with genetics. So like if your mother, father, aunt, uncle, or cousin, brother, or sister have this specific type of schizophrenia, that means that you have a higher risk of having it yourself or of getting it later on in life. And this is associated with a disproportionate number of summer births. Now remember I said the non-deficit type has a higher correlation with winter births and is associated with flu epidemics. So that winter type, or this non-deficit type, this typical, we'll put typical type, is associated with more with environmental factors. So the way I'm going to remember this, of course, my little brother, he has both positive and negative symptoms. He has this grandiose vision of who he is, and he also has this this disconnected world that I have no idea what it is or what's going on in this world that he's in. But he also has these negative things, like he's addicted. He smokes. He has a lower activity and flat speech affect. So he has this, what you would consider a typical type, correlated with environmental factors. What I'm trying to say, guys, really is that I'm probably not crazy. Okay? Because it doesn't run in the genes. So here's what are the first-ranked symptoms of schizophrenia, and I'm not just going to read them all to you, but a couple of them I want to point out. My little brother, whenever I first realized there was definitely something wrong with him, he came up to me and he was like, "I know you know what I'm thinking." He's like, Some of us can tell what other people are thinking. You can hear what I'm thinking, right?" And I was like, "No, but I'd be really glad for you to tell me." He's like, "Don't pull my chain." So these are first-rank symptoms, and they're important in the diagnostic algorithm of how do you know if somebody has it. So you have to have 2 first-ranked symptoms and 2 second-ranked symptoms or a deficit symptom. And so you look in the DSM-5, it's a complicated, like you have to go through and check off each thing, and that's how you diagnose. You can just go through your symptoms of first-rank, second-rank, deficit. You check off the things, and if it meets all the standards and criteria and you ruled out everything else, then it's schizophrenia. So what do you want to rule out? The first thing you want to rule out is Wilson's disease. So you would look for the Kaiser-Fleischer rings in the eyes, and so this would be a problem with copper metabolism. You could check ceruloplasmin as well. The differential diagnosis for psychiatric disorders this would be like a metabolic disorder, Wilson's, I'm pretty sure. But the psychiatric disorders you want to rule out would be schizophreniform disorder, schizoaffective disorder, bipolar, major depression with psychotic features, and substance abuse psychotic disorders. And with my little brother, I think actually this is the only one that wasn't ruled out, and there's some more background information on why I think that's important, but that's personal. Okay, we're going to change gears a little bit because in our case we had some protein and epithelial cells in the urine, and so that's my objective I'm working on, as well as the schizophrenia. So what is the protein in the urine? It's typically an indicator of glomerular or tubular renal dysfunction, or function rather. So are the glomerulus filtering out the protein? Yes or no? And then if they are, is there another way for protein to get into the urine? Now, that was my personal question. I couldn't find anything that answered that definitively, but I thought perhaps that cell lysis could leak protein into the urine, and I didn't find that anywhere. So, but here are the lists from the various differential diagnosis books. They all list basically the same thing. So nephrotic syndrome, preeclampsia is pretty big for pregnant women. If you have protein in the urine, that's the very key indicator that of preeclampsia, and so they got to start getting evaluated, checking their blood pressure and that kind of stuff. And of course, diabetes mellitus, we've all become very familiar with the fact that high blood sugar can cause damage to the kidney. Glomerulonephritis, amyloidosis, which is the deposition of amyloid plaques at various places. There's different types of amyloids, and that's something else that can cause this damage. And then another one would be multiple myeloma, is on the differential with protein in the urine. Okay, now protein in the urine, what you want to do if you have that is you want to get a 24-hour urine specimen to quantify the amount of protein. That'll give you an indication of the severity of what's going on. But and that's usually what you would do. A, whenever you know the diagnosis, and B, so when you know the diagnosis, you do that. But you can also do that without knowing the diagnosis, I guess, just to check on renal function in general and to treat that accordingly. But there are some interfering factors, so some things that can cause increased protein in the urine. And one thing that I found was really interesting is that if you take a cold bath, it will increase the protein in your urine. I'm not sure exactly how that works, but I'm guessing it has. I have no really good guesses, so your guess is as good as mine. Radiopaque contrast. If you've got a contrast imaging within the last 3 days, you're going to get a false positive on the proteinuria. And then, of course, there's contamination from the prostate or the vagina, so different things can cause secretions there, and I don't want to go into that because they're gross. The high protein diet, if you're working out and you're taking 82 grams of protein every 3 hours, so you get huge muscles like mine, then you'll have also protein in your urine. Now, if you're measuring protein using the dipstick method, there's something called Bence-Jones proteins that can show up there. And what that's suggestive of is multiple myeloma and Waldenstrom's macroglobulinemia. And I highlighted Waldenstrom's because I looked into the symptoms, and several of them fit with symptoms we have in our case. I don't think it's really high on the differential because a lot of the symptoms would occur, a lot of the symptoms where we're seeing parallels in our case would occur very late in the disease process, and we would see there's a lot of other things that we should see with Waldenstrom's that we don't, but it was out of all of the various things for protein in the urine, this was the only one that really jumped out with its list of symptoms and such. Other things that can cause protein in the urine, it got cut off here as some drugs, and there's a list of medications, and I didn't want to put them all on here, but a list of medications in the study guide you can go look through, and it can cause proteinuria. What about the epithelial cells? Well, you have desquamation, so the cells will typically, as the basal layer of the epithelium is growing from the bottom, it's pushing all the older cells up, and they desquamate, and that would be typically the cells of the bladder and urethra. When kidney cells desquamate, typically they'll form casts, and so you see those epithelials less often in a urinalysis, but it's not completely impossible. So then what are the abnormals? Usually you get reported as few, moderate, or many, and I couldn't find a table to say how that's calculated, if a few is 1 to 15 or whatever the case may be. So I wasn't able to find that, but it's usually reported as few, moderate, or many. In our case, it was moderate at low power focus. And it will typically be indicative of an inflammation, infection, or malignancy, or the other possibilities that it's contaminated from surrounding tissues. So if cells from the tip of the penis, for example, got into the urine sample, then that would cause a contamination. Another thing we didn't see on our report was the type of epithelial cells, and everywhere I looked said you should know the type of epithelial cell because it will help you to localize the problem. For example, we know that in much of the urinary tract, it's lined with something called transitional epithelium. So transitional, and if we knew that we had transitional cells, we would at least know that a larger portion or specific portions of the urinary tract could be affected. If it was something like cuboidal, or for example, cuboidal cells, then we would be looking within the kidney or that type of thing. Now we've all studied histology, so go back and look at your histo notes, and then kick whoever wrote this case up for not being more specific.